Hello, today our presentation will review medium-chain acyl-CoA dehydrogenase, otherwise known as MCAD, and MCAD deficiency. MCAD deficiency is a genetic disorder that affects mitochondrial fatty acid oxidation in humans, resulting in high blood levels of fatty acid intermediates and hypoglycemia. MCAD is a flavoprotein found in the mitochondrial matrix that acts on 4 to 12 carbon chains that catalyze the alpha-beta dehydrogenation of fatty acid acyl-CoA conjugates. MCAD interacts with electron-transferring flavoproteins, aka ETF, in order to enable successful flavin-to-flavin electron transfer, crucial for the beta oxidation of fatty acids. MCAD is a tetramer made of 4 identical subunits with 2 active sites which work independently from one another. The substrate FAD acts as a cofactor which helps maintain the stability of MCAD as well as the volume of the enzyme's catalytic binding site. Moreover, the N-terminal and C-terminal domains are composed mainly of densely packed alpha helixes, and substrate fatty acyl moiety binds to the active site between helix E and G, and is lined with the side chains of glutamic acid 376, tyrosine 375, valine 259, threonine 168, leucine 103, and alanine 100. Binding between the flavin ring and the carboxylate of glutamic acid 376, several tightly bound ordered water molecules at the active site are displaced. An alkyl chain lies in the substrate cavity surrounded by glutamic acid 99, threonine 96, and tyrosine 375. The tyrosine surrounds the tail of the alkyl chain and conserves sequences of acyl-CoA dehydrogenase, which plays a role in the binding of the substrate.
Beta oxidation overview. Beta oxidation is a sequential 4-step reaction. In these 4 steps, fatty acid molecules go through oxidation, hydration, oxidation, and cleavage respectively. At each cycle of 4 steps, 2 carbon units are removed from fatty acyl-CoA molecule. The removed 2 carbon molecules, acyl-CoA, can go and participate in DCA, and the remaining fatty acyl-CoA molecule goes back in the beta oxidation reaction. This cycle continues until the fatty acyl-CoA molecule is completely converted to acetyl-CoA. Metabolic pathway of fatty acid in MCAD deficient patients. In MCAD deficiency, the long chain of fatty acid goes through the beta oxidation cycle until the fatty acyl-CoA chain reaches C6C12N. However, in the absence of MCAD, further breakdown of fatty acyl-CoA chain does not occur and it starts accumulating in mitochondrial matrix. The accumulation of fatty acyl-CoA causes a toxic effect on the inner mitochondrial membrane. As a result, fatty acyl-CoA move out into the intermembrane space. Once in intermembrane space, there are 2 possible fates of medium-chain fatty acyl-CoA. Firstly, the enzyme ZPT1 attaches fatty acyl-CoA to the carnitine and converts it into fatty acyl-carnitine. The medium-chain fatty acyl-carnitine will then be pushed out into the cytoplasm, and from there it will enter the bloodstream. Secondly, C6C12 fatty acyl-CoA can diffuse through the outer mitochondrial membrane into the cytosol. It enters the endoplasmic reticulum, which converts medium-chain fatty acyl-CoA into dicarboxylic acid, and from there, it enters the blood. Now let us see how metabolic control is regulated and how it could be affected by MCAD.
Fatty acids will enter the intermembrane space via fatty acid transporter. Using fatty acyl-CoA synthase, a CoA group is added to create a medium-chain acyl-CoA. CPT1 then transforms medium-chain acyl-CoA to a medium-chain acyl-carnitine. MCA is now passed along the inner mitochondrial membrane. CP2 picks it up and converts MCA back to medium-chain acyl-CoA. MCAC can now undergo beta oxidation, which will make 1 acetyl-CoA for each cycle, bringing it to the Krebs cycle. Adding on to what was mentioned in a previous slide regarding how MCAD affects the metabolic pathway, these increased levels of acetylcarnitine and decarboxylic acid serve as a diagnosis marker. Therefore, profiling of blood and urine by tandem mass spectrometry is done to determine FAO's genetic defects, especially in the newborn. Metabolism at a tissue level is controlled by insulin and glucagon. Insulin will be be released when blood glucose levels are high. It will promote the uptake of glucose in the muscle and adipose tissues and will trigger glycolysis and turn off gluconeogenesis. Glucagon will be released when blood glucose levels are low. Its main job is to increase CAMP, which creates a cascade of phosphorylations of key enzymes such as phosphorylase, beta to alpha, and the other three pictured in the diagram. The above four reactions increase blood glucose levels. Both insulin and glucagon are key to managing blood glucose levels and the generation and use of ATP. When hormones signal for the need of stored energy, tags are released from adipose cells and transferred into various tissues where fatty acids can be oxidized to produce ADP. The hormone epinephrine starts this cascade of reactions to remove one fatty acid at a time until there's one glycerol and one free fatty acid. The one free fatty acid is transferred into the blood by serum albumin.
The MCAD enzyme is coded by the ACADM gene, which is located on chromosome 1 at position 31. MCAD deficiency is caused by a variety of mutation types, but the most common type of mutations for this disorder are point mutations. Point mutations occur when one base is replaced by another in the DNA sequence. The most prevalent mutation occurs during DNA replication when the 985th base, an adenine, is replaced by a guanine. The base change results in a change from lysine to glutamic acid at amino acid 304 during translation. Because of this change, this mutation is referred to as the K304E variant. MCAD is made up of 4 identical subunits called a tetramer. The result of the K304E mutation is that the tetrameric structure of the enzyme is unable to form. This occurs because of the side chains of lysine and glutamic acid. Lysine 304 is part of one of the alpha helices that form part of a subunit. Lysine has a positively charged side chain, while glutamic acid is negatively charged. The result of this is that the negative side chain of glutamic acid repels aspartic acid 346 on another helix. What occurs is a distortion in the monomer or subunit that makes an unfavorable surface for another subunit to dock. Thus, the enzyme is unable to form properly, impairing function.
The main vitamins and minerals that are of importance in MCAD is riboflavin. Flavins are protein cofactors that participate in oxidation, reduction, and dehydrogenation reactions. Acetyl-CoA is formed and transferred into the mitochondrial membrane as a derivative of carnitine when it is activated within the cytosol during mitochondrial beta oxidation. A dehydrogenation then occurs to the acetyl-CoA fatty acid. Acetyl-CoA dehydrogenases are flavin-containing enzymes that help derive electrons from the dehydrogenation reactions, which are accepted by an electron transfer flavoprotein. FAD is also present to help MCAD fold into its final structure. Riboflavin deficiency can have major implications on the metabolism of fatty acids, as FAD is a cofactor in fatty acid beta oxidation. One study found that riboflavin treatment was able to raise residual MCAD activity from 5.2% to 19% since FAD stabilizes MCAD. When ETF activity is decreased due to riboflavin deficiencies, the activity of acetyl-CoA dehydrogenases is also decreased, and MCAD activity in particular is decreased by 65% by just the second day of the deficiency. MCAD was first identified in 1976 and is the most frequently occurring fatty acid oxidation disorder. MCAD is most prevalent in Europe, where the frequency of diagnosis is 1 in 5,000 to 1 in 20,000, and is the only fatty acid oxidation disorder that is approved for newborn screening. Usually, MCAD presents itself in children between the ages of 2 to 3 years old. 17% of children with MCAD will most likely die from it, and 10% will go on to have severe disabilities. Symptoms of MCAD deficiency include low blood sugar, vomiting, and fever, which can be worsened by prolonged fasting periods. However, riboflavin supplementation can help improve MCAD deficiency. Riboflavin is also added to baby foods and cereals, while other sources include milk, fish, and eggs.